Clinical Chemistry, Introduction to Carbohydrates. Carbohydrates can be classified based on several different factors, one of them being the size of the base carbon chain. A triose has 3 carbons, tetrose has 4, pentose has 5, hexose has 6, and there's some representative diagrams below. They can also be classified based on the location of the CO function group. An aldose has a terminal carbonyl group, or an aldehyde group. In a ketose, the carbonyl group is in the middle, and it's linked to 2 other carbon atoms, a ketone group. So here you can see what a ketone looks like, with the R's being 2 other groups attached, and this is an example of a ketose. An aldehyde you can see with the terminal carbonyl group, and here you can see the aldose. It can also be classified based on the stereochemistry of the compound. There are 2 different series that are possible, a D and an L, and here you can see that they're mirror images. So L-erythrose and D-erythrose are mirror images from each other. So you can see the OH over here and the OH over here, so how they are flipped. So this would be the L configuration and this is the D configuration. They can also be classified based on the number of sugar units in the chain. Monosaccharides have 1 unit, and examples of monosaccharides include glucose, fructose, and galactose. Mono means 1, saccharide is referring to the sugar. Disaccharides, di means 2, so di has 2 units. Maltose, lactose, and sucrose are examples of disaccharides. Poly means many, so polysaccharides have greater than 10 units. Starch and glycogen are examples of polysaccharides. Carbohydrate metabolism. So the fate of glucose depends on the metabolic needs of the body. So energy production is one of the major reasons why we need carbohydrates coming into the body. So we convert to that sugar to carbon dioxide and water. We can also store the glucose in the body, so we can store it as glycogen in the liver or as triglycerides in adipose tissue. Conversion to keto acids, amino acids, or proteins. And CHO or carbohydrate metabolism is interwoven with that of lipids and amino acids. So when dietary carbohydrates are ingested, salivary amylases start to hydrolyze the starch. Salivary amylase is inactivated by gastric hydrochloric acid though, so once it gets to the stomach it is inactivated. Carbohydrate digestion will continue once it gets to the duodenum. Pancreatic amylases hydrolyze partially digested polymers into intermediate-sized glucosans, and then it's further digested in the small intestine by maltase, sucrase, lactase, and other such carbohydrate digesting enzymes until monosaccharides are formed. Monosaccharides are then going to be absorbed by the intestinal lumen and transported through the bloodstream to the liver. If we start with glucose here, glucose comes into the cell and is converted into glucose 6-phosphate. Once it's converted into glucose 6-phosphate, it's kind of trapped inside of the cell, so that's how we get glucose to stay inside of the cell. Glucose 6-phosphate then has multiple pathways that it can take depending on the needs of the body. If we don't need a lot of energy at the moment, or if our cells don't need to go through the process of cellular respiration, glucose 6-phosphate can be converted to glucose 1-phosphate, and that can start the process of storing glucose in either the liver or the muscles as glycogen. So we can go through the process of glycogenesis to create glycogen. Now, a lot of these terms are going to sound very similar, so I encourage you to break the terms down into their stems. Glyco is talking about glycogen. Genesis is the creation of something. So glycogenesis is talking about the creation or the formation of glycogen. And glycogen is our storage form of glucose. Like I mentioned, glycogen is going to store glucose in the liver or in our muscle tissue. If we suddenly have a need for glucose, we can take that glycogen and break it down to convert it back to glucose 1-phosphate, back to glucose 6-phosphate, and then we can use it in cellular respiration. That process is called glycogenolysis. Glycogen is in the word. Lysis means to break apart. So glycogenolysis is to break apart. Starting back with glucose here, we have glucose that goes to glucose 6-phosphate, and then this can enter into the uronic acid pathway to produce glucuronic acid. It can also go to the hexose monophosphate shunt to create ribose and carbon dioxide. It can also more commonly go into the cellular respiration cycle. So we have glucose 6-phosphate through a number of steps, 10 steps in glycolysis, are going to convert that to pyruvate. Pyruvate can then go through the tricarboxylic acid cycle, the TCA cycle, also known as the Krebs cycle or citric acid cycle. And through that process, it's going to then go through oxidative phosphorylation in the electron transport chain and be converted to adenosine triphosphate or energy. Pyruvate can also, if we don't need to produce energy, it can be converted back to glucose. So gluconeogenesis. Gluconeogenesis. Gluco is referring to glucose. Neo means new. Genesis is the creation. So we're creating new glucose from a molecule from some other molecule, so not a carbohydrate, gluconeogenesis, to produce glucose again. So this is kind of a good summary table to review. These are some definitions for those of you who like definitions written out. So glycolysis, glucose to pyruvate. Glycolysis is kind of how we more commonly say this, but like I said, I want you to break these terms down into their stems. So glyco is talking about glucose, lysis means to break apart. So we're literally breaking apart glucose to form pyruvate. And glycolysis is that first phase of cellular respiration. Gluconeogenesis, we're producing new or creating new glucose from non-CHO sources. This is going to account for maintenance of blood glucose with prolonged fasts. Glycogenolysis. Glycogen lysis means to break apart glycogen. So glycogen is broken down to glucose for energy maintenance, and this is with brief fasts. And glycogenesis is when glucose is converted to glycogen for storage. This is an interesting chart showing the sources of blood glucose and how they are impacted throughout the day. When labs are being drawn and when we're evaluating specimens for levels of glucose and different hormones that are going to regulate glucose, this is something to keep in mind, that glucose levels are going to be continuously fluctuating throughout the day, and different sources are going to contribute to that glucose at different times of the day and depending on when the patient has eaten or not. So part of the sampling considerations that they're really important for when measuring blood glucose levels are going to be fasting status. You don't need to memorize all of these different peaks here. This is just something interesting to look at. So kind of reiterates the point that different times of the day are going to be impacted in different ways by blood glucose levels. So the regulation of carbohydrate metabolism, the liver, pancreas, and other endocrine glands control blood glucose concentrations within a narrow range. There are several hormones that control glucose levels, and this is just a brief summary. I am going to go into a lot more detail of each one of these hormones as we go through the rest of the slides for this section. So insulin is produced in the pancreas. It's going to decrease blood glucose levels. The main targets of insulin are going to be the liver, skeletal muscle, and adipose tissue. Glucagon is also produced in the pancreas, and it increases blood glucose levels. Epinephrine and glucocorticoids are produced from the adrenal gland, and they increase blood glucose. Growth hormone and adrenocorticotropic hormone, or ACTH, are produced in the anterior pituitary, and they increase blood glucose levels. Thyroxine, or T4, is produced by the thyroid gland, and somatostatin is produced by the pancreas, and they both increase blood glucose levels. So insulin is a 6 kilodalton protein. It's produced by beta cells of the islets of Langerhans in the pancreas. It is an anabolic hormone which stimulates the uptake of glucose into fat and muscle tissue. It promotes the conversion of glucose to glycogen or fat for storage. It inhibits the production of glucose by the liver. It stimulates protein synthesis, and it inhibits the protein breakdown. The synthesis of insulin is important to make note of because some of the defects when we talk about diabetes mellitus have to do with errors in the synthesis of insulin. So it is synthesized as pre-proinsulin. It's enzymatically cleaved and converted to proinsulin. Proinsulin is then stored in secretory granules in the Golgi of the beta cells. 10% of the activity of insulin, and it is the major storage form of insulin. Proinsulin is cleaved to insulin at the C-peptide. Insulin and C-peptide are released in equimolar amounts into the portal circulation. C-peptide seems necessary to ensure correct structure of insulin. It's removed and excreted by the kidney. Glucagon is a 29 amino acid polypeptide that's secreted by the alpha cells of the pancreas. The major target is the liver, and a minor target is adipose tissue. It increases lipolysis. Lipo is referring to lipids. Lysis means to break apart. Secretion is regulated by plasma glucose levels, and it's also stimulated by stress, exercise, and amino acids. It is inhibited by insulin. There's an impaired response to hypoglycemia with long-standing diabetes mellitus. High concentrations contribute to hyperglycemia and ketosis of diabetes. Epinephrine is a catecholamine that's secreted by the adrenal medulla. It stimulates glycogenolysis, so again, glycogenolysis to break apart glycogen, and glucagon secretion. It decreases glucose use, insulin secretion. Secretion is impaired with type 1 diabetes. Key role for glucose counterregulation with glucagon. Regulation of lipid metabolism, glucagon and epinephrine activate hormone-sensitive lipase in adipose tissue. In coordination with the activation of gluconeogenesis in the liver, metabolism of fatty acids through beta oxidation in the liver yields ATP for gluconeogenesis. Acetyl-CoA is converted to and released to blood as ketone bodies. If you remember the function of epinephrine, epinephrine regulates the fight or flight response. In order to be able to fight or run away, you're going to need a lot of energy. So it's going to want to mobilize as much glucose as possible to have that energy that is needed for the fight or flight response. One rich source of energy is going to be adipose tissue. So it's going to start breaking down adipose tissue to convert that into the glucose through the process of gluconeogenesis. Other hormones, the growth hormone is a polypeptide that's secreted by the anterior pituitary. It stimulates gluconeogenesis and lipolysis. Again, if you're going to be growing, you're going to need energy in order to do that, and energy is going to be produced by the breakdown of fat and conversion of the fat to glucose. Cortisol is secreted by the adrenal cortex in response to adrenocorticotropic hormone. It stimulates gluconeogenesis and increases the breakdown of protein and fat. In Cushing's syndrome, hyperglycemia is noted due to an increased production of cortisol by a tumor or hyperplasia of the adrenal cortex. In Addison's disease, you have hypoglycemia due to the loss of cortisol that's caused by the destruction or atrophy of the adrenal cortex. Thyroxine, or T4, is secreted by the thyroid. It's not directly involved in glucose homeostasis. It stimulates glycogenolysis and increases the rate of gastric emptying, intestinal glucose reabsorption. It may produce glucose intolerance in thyrotoxic individuals. Somatostatin, or growth hormone inhibiting hormone, is found in the GI tract, hypothalamus, the delta cells of the pancreas. It has no direct effect on carbohydrate metabolism, but it inhibits the release of growth hormone, glucagon, and insulin by the pancreas. And this is a good diagram that kind of puts this all together. So here you can see the pancreatic cells, so alpha, beta, delta. The beta cells are going to release insulin, and insulin is going to act on lipids to cause glucose uptake. So lipogenesis, it's going to cause the creation of fats from those glucose precursors. And you're going to have glucose uptake into the adipose tissue. Insulin is also going to act on muscles so that we have increased uptake by the muscles. Here you can see the alpha cells are going to produce glucagon, and glucagon is then going to act on the liver to cause gluconeogenesis and glycogenolysis. We also have the growth hormone and cortisol that are also going to act here, and epinephrine is going to act here as well. And somatostatin is a negative regulator of both the alpha and beta cells, so it's going to stop production of both.